And our third speaker in this section is Rachel Floyd, who's just finished, I think, her postdoctoral fellowship, Queen of Urinary Tract Infections, based in Liverpool.
So, forwards, backwards. Oh dear, you're not doing, sorry.
Thank you. So, I've reached the end of my fellowship, and it occurred to me while I was thinking about this research area that some of you will have actually heard me talking about the same thing for the last 3 years. So I thought, well, I'll take a completely different approach and I'll tell you about some of the new research we're doing in my lab at the moment. So some of the stuff we've been doing has been looking at this very woolly area. In truth, our real research aim is to understand whether hormones make bugs bad. Because urinary tract infections are incredibly common, so lots of people get them, and lots of people recover spontaneously. So many of them will recur spontaneously with no treatment. Many of them can be treated very effectively with antibiotics. But there are some infections that don't respond very well to antibiotic treatment for a number of reasons. So we're interested in understanding what drives these infections. So the main area of research within my lab is to understand the pathophysiology of urinary tract infections. How do they arise and what makes them worse? So UTI can be broadly classified into two different types. So uncomplicated UTI generally occur in women of childbearing age with no other confounding factors, so they're otherwise healthy. They can occur in people of all ages, but the primary group that is affected are women of childbearing age, and they generally don't cause too many complications. The other classification of UTI are these complicated UTI, where there is some other abnormality that causes problems. So it's generally something like a structural abnormality, like a stricture or catheterization, metabolic abnormalities such as diabetes or renal failure, or impaired host response, so in patients with HIV or patients who are on immunosuppressive medication. And this is associated with an incredibly high mortality rate. So we're clearly not doing a very good job, one, at diagnosing and preventing these infections, and two, at treating them when they actually occur. So we really want to understand more about these complicated UTIs and what's driving them to be more aggressive, more drug resistant, what is actually going on in the pathophysiology of these infections and how can we alter that? So if we look at the types of bacteria that cause uncomplicated UTIs, most of them are caused by uropathogenic E. coli. And it can be up to 90% of them are just caused by this one family of pathogens. When we look at complicated UTI, there's a much broader spectrum of families of bacteria that cause these infections. So we really want to understand why these guys are suddenly getting a much bigger piece of the pie. Why are they outcompeting uropathogenic E. coli? And one of the bacteria we're particularly interested in my lab are Pseudomonas aeruginosa. So you can see in this red fragment here, because 3 times more complicated UTIs are caused by Pseudomonas. And there's a number of reasons why this guy is particularly interesting. So it's an opportunistic pathogen. And basically what that means is it's a bacteria that takes advantage of anyone with any immunocompromisation or any sort of abnormality structurally or functionally. And these guys are incredibly resistant to antibiotics inherently. So they're genetically pre-programmed to resist the effects of a number of antibiotics. They also express a lot of virulence factors. So these can be considered to be the angry factors. They're the things that make bacteria really good at causing disease. Now we say they've got a versatile metabolism. Basically what that means is they'll grow on anything or in anything. They'll grow in soil, in water, on hard surfaces. They really don't care. And this is why there's such a huge problem in healthcare settings. So Pseudomonas cause 1 in 10 of all hospital-acquired infections. One of the other reasons Pseudomonas are so good at causing these infections and so difficult to eradicate is because of biofilms. So the easiest way to explain what a biofilm actually is that it's a sugary igloo that protects the bacteria inside from any cleaning agents, any abrasion, or any outside environmental stresses. So basically what happens is you have bacteria that will attach to a surface. So this could be a catheter, it could be a worktop or a piece of equipment in a hospital. And the bacteria will begin to synthesize this matrix. And within this matrix, they're protected from any sort of external stress. So they can grow quite happily inside this biofilm. Also, one of the problems we have is that within the biofilm, the bacteria start to evolve. So they change, their genetics change. So one population of bacteria within this biofilm will be very similar to the initial invading bacteria, and the other will have developed an antibiotic resistance that is 1,000 times greater than the original invading strain. So that is a huge problem when you think of this in a clinical context. If you've got these subcolonies of bacteria that are growing quite happily, you can't wipe them away, you can't clean them, and you can't kill them. So what exactly do we need to know about these guys? Well, if we can understand what causes these processes to occur and makes them happen more quickly, we're in much better position to be able to treat these infections more effectively. So if you think of every single infectious disease as a series of molecular conversations between host cells and pathogens, we want to understand if there's something that the host cells are saying that is making the bacteria really angry. What are they doing that is making bacteria so much better at causing infections? So we're trying to understand if there is a chemical signal or signals that's coming from the host cells that makes the bacteria really angry and good at causing disease. Now, if you look at the literature, there is actually a huge sexual dichotomy that exists in a number of infectious diseases, whereby clinical outcome is actually far worse in either males or females. So with malaria, cystic fibrosis, measles, shingles, and HIV, clinical outcome in women is generally much worse. And conversely, for men, TB staph and group A strep and leptospirosis and meningitis, men do far worse when they're infected with these diseases. So it suggests there might be some hormonal influence that's driving this sexual dichotomy that we see. So we wanted to understand whether that could exist in urinary tract infections as well. And when you look in the literature, you find out that in a normal menstrual cycle in a woman, where estrogen peaks at day 14, women are 3 times more likely to experience a urinary tract infection at this point in time. So there's clearly something going on at this point within the body that is causing bacteria to become more aggressive or better at causing infection. So we wanted to understand a bit about what might be going on. So we decided to look at some of the key traits of bacteria that make them really good at causing infections. So we've used an artificial urine model that's spiked with hormones to analyse each of these different characterisation traits. And we've looked at 20 different isolates of bacteria from patients with complicated UTIs. So the first of these parameters is growth, because growth will dictate how fast bacteria can spread, how many people they can infect, and how they can get from A to B more quickly. And surprisingly, when we actually looked at growth of these bacteria in the presence of hormones, it slowed down, which is completely the opposite to what we thought might happen. So it suggests that hormones are actually protective against the spread of Pseudomonas infections, at least in urinary tract isolates. So the next thing we wanted to understand was about how bacteria stick around in these biofilms. Can hormones make these biofilms form more quickly? Can they get bigger as a result of hormones being there? What is actually going on? So we used a dye that will actually bind to these biofilms so we can find out how big they are. And what we found is that in the presence of hormones, so progesterone, testosterone, and estrogen, the biofilms are much smaller. So again, hormones are having a protective effect in preventing colonization by Pseudomonas. So one of the questions we raised was whether this response is different if you get bacteria from male patients or female patients, because they will have seen a different hormone profile in their host. And the answer is simple. No, there is absolutely no difference in response whether the bacteria come from a male patient or a female patient. hormones respond in exactly the same way in this system. So the next thing we wanted to understand is how is resistance to antibiotics potentially affected by the presence of hormones? Because this is a rising problem at the moment. There's a huge issue with resistance to multiple antibiotics, and Pseudomonas, as we've already said, is already inherently resistant to lots and lots of antibiotics. Now, if you look in the literature, there is evidence that estrogenic compounds, or mimics of estrogen, can actually cause an upregulation of antibiotic efflux pumps. So these are pumps that sit in the membrane of bacteria and pump antibiotics back out. So it makes the bacteria resistant to the effects of antibiotics. So they can't be killed. So the more of these pumps you've got, the more resistant to antibiotics bacteria are. So it led us to ask the question whether hormones can make bacteria more resistant to antibiotics by increasing the number of these pumps. So we wanted to look at clinically relevant antibiotics. Now, one of the problems we have is that Pseudomonas are completely resistant to these two, and coamoxiclav, and about 50% of isolates are resistant to gentamicin, and that figure is rising. So clinically, we're left with meropenem. And what we've actually found is when you apply increasing doses of hormones, you get a switch in resistance profiles in the bacteria. So strains that once were sensitive to meropenem suddenly become completely resistant to the effects in the presence of progesterone, estrogen, and partially in testosterone as well. So there's a dose-dependent shift in antibiotic sensitivity in the presence of hormones. So it suggests that the hormones are actually doing something to the bacteria to make them more resistant to the effects of antibiotics. So the final thing we wanted to address is these angry factors, the things that make bacteria really good at doing what they do. So these are proteins that help bacteria to stick to their host cells, invade, create toxins that help them to outcompete other bacteria. So we wanted to understand how hormones influence these virulence factors. Now, in Pseudomonas aeruginosa, the transcription of all of these virulence factors is regulated by a global regulator called LASR. So LASR will actually switch on transcription of all of these angry factors. So we wanted to understand what hormones do to that process. So we've only looked at progesterone so far because all of this is very preliminary work, but we've actually found that there's a threefold upregulation of expression of this gene in the presence of progesterone. So that suggests to us that the presence of this hormone is actually making these bacteria more aggressive and more capable of invading and surviving in host cells. So collectively, what does this data really mean in the bigger picture? What does this say to us? Well, although the effects of hormones seem to be making the bacteria grow more slowly so they can't infect quite so many cells, and it also makes them less persistent because they don't form as many biofilms, the bacteria that are there are incredibly aggressive because they've got lots of virulence factors, and they're also increasingly resistant to antibiotics. And this is a classic trade-off that you see in bacteria, where fitness in terms of replication is sacrificed to be able to survive in the host. So in the grand scheme of things, this actually means that if we understand the processes that are driving resistance and persistence in these bacteria, we're in a much better position to be able to treat these infections more effectively. And it also gives us the option for a personalised medicine approach, because if we understand that there are people who are likely to be susceptible to these types of infections or are infected with a hormone-sensitive strain of bacteria, it means that we can approach the treatment much more effectively than previously we have. But this is obviously a long way in the future, so this is very preliminary work. So I'd just like to finish by thanking the people who actually did the research that I've just highlighted. So this is Christina, my research assistant, Salma and Dorota, my MRes students, Amy and Charlotte, who are my undergrad students. This is Joe Fothergill, who's an infectious disease researcher at the University of Liverpool, and John, who's our PhD student, who's going to be carrying on this work for the next 3 years. I'd also like to thank Mersey Kidney First, who are funding John's PhD, and obviously to these two charities who funded this pump priming research. And I'd like to say a massive thanks to Kidney Research UK, because without your support and also all of the additional development opportunities you've given me, I wouldn't have been able to establish my own lab and reach this point in time where I do have my own line of research to show for it. Thank you.
So wonderfully clear, as always. Where does the oral contraceptive pill fit into this story?
It's a fabulous question, and it's one that we're looking at the moment because there's evidence in a lot of different studies. So postmenopausal women are incredibly susceptible to UTI. And there's a difference in susceptibility based on whether you are on hormone replacement therapy or not. So one of the avenues that we're actually taking in the next few months is to apply for funding to look at whether there is any correlation between women who are on contraceptive pills and their susceptibility to urinary tract infections. We're also going to correlate that with infections in pregnant women. Because we're based at Liverpool Women's Hospital, we're obviously very interested in understanding, because pregnant women get a lot of these types of complicated UTIs, whether there's a difference in their profile as well in terms of susceptibility and the types of bacteria that we isolate from these infections. It also leads on to another problem with environmental pollution. So many of you will already be aware that contraceptive pill is found in the watercourses globally. And a lot of these bacteria, so Pseudomonas, are inherently present everywhere in the environment. And if you have that selective pressure where there's constant exposure to contraceptive pill hormones, it suggests that you might be driving this level of super resistance in the environment that can then be transferred to humans.
Right.
A little bit worrying, isn't it?
You show that with the three different hormones, estrogen, testosterone, progesterone, you see typically actually the same effect, whether it's increased resistance or decreased bioinformation. Have you actually tried to mix them?
We haven't. It's something that one of my students is starting to do. So we've got an intercalating medic that's starting in 3 weeks, funded by Kidney Research UK again, and that's something she's going to be looking at. And she's also going to be looking at co-infections as well, because obviously the virulence factors that Pseudomonas express depend very heavily on whether it's a mixed infection or a single bacteria infection. So it's a very good question. It's something
And
we hope to look at.
I don't know, would you have expected that all those three hormones actually behave in the same way?
No,
Isn't
we
that
didn't.
surprising a little bit?
We didn't expect them all to behave the same way. You do see a dose-dependent effect. So progesterone always seems to be the most aggressive. And of course you get progesterone in men as well. So there's obviously going to be overlap between a lot of these responses. And the profiles of these hormones change very much between people and change with age as well. So I'm sure there's also immune factors that are playing a role as well, because you see differences in immune status between men and women. So there's going to be complex interplay between all of these factors as well. It's not just going to be down to hormones.
Yeah. Thank you.
Pseudomonas often colonizes the lungs of people with CF.
Yes.
And I wonder whether there are any differences in the outcomes or sensitivities of resistance in patients with CF according to gender, because they have a
Yes,
different
there are.
hormonal milieu and they've got the same anatomy as well.
Yeah. So women generally do much more poorly if they're infected with strains of Pseudomonas than men do. So the slide that I showed you where it suggests that there's upregulation of efflux pumps, that's actually research that's been done on Pseudomonas isolates from a CF patient.
Is there any difference in resistance profiles in women that get these infections pre and post-menopausally?
We don't know. It's something we'd like to look at, because it's obviously something that's really interesting based on a hormone profile that the bacteria will have been exposed to. So I think it's a really interesting question, because this is it's surprisingly, nobody has ever looked at this area of research before. We were quite shocked when we looked into the literature and thought, well, obviously there's differences in hormones. Women get a lot more infections than men do, but it's always been attributed simply down to anatomical differences. But it seems that there's a lot more complexity to it than just that, because of this complicated UTI situation where you've got women and men are both getting similar numbers of infections. So there's something else going on there.
So there's plenty to do, isn't there?
Yeah, definitely.
Just what about the pre and post-pubertal state? I'm particularly with Jeremy's example of CF. So is there a difference pre-pubertally and post-pubertally in Pseudomonas?
Sorry, can you repeat?
Is there a difference between pre-pubertal and post-pubertal patients in terms of their response to Pseudomonas in CF?
I don't know. It's one of my colleagues, Jo Fothergill, is actually working on that right now. So she's been looking at exacerbations and looking at whether there are differences in responses to hormones in these patients, depending on which point in time the isolates have actually been taken from patients. So there's a lot of evolution that goes on in Pseudomonas. So a patient can be infected with a single strain and they keep that strain for life, but the strain constantly evolves due to different changes in environmental stresses.
Any more for him? Okay. Oh, yes.
One at the back.
I was just wondering where you thought the sort of sudden spike in the urinary tract infection over that sort of 14-day period. Is that just bacteria that are present and then just switching on virulence factors due to the hormonal change, do you think, or is it additional colonisation just because of the change in the biofilm? You'd expect that maybe that it's not additional colonisation, it might just be that.
I think there's a lot more factors to it, because there's a lot more changes in vaginal microflora as well. So you get changes in lactobacilli that also compete in these types of infections. So I think, again, it's driven by a lot of different factors. So I think that spike partly may be due to the spike in estrogen that we're seeing that affect bacteria, but it's also down to the fact that maybe bacteria have an easier time colonizing in the first place because of changes in microflora elsewhere.
Great. Thank you very much, Richard.
Thank you.